In this video, we are going to discuss about affinity chromatography. Okay, so affinity chromatography is the purification of the biomolecule, and this purification of the biomolecule will depend upon the biological function of the biomolecule or the individual chemical structure of the biomolecule. Okay, the separation is done on the basis of the properties of this biomolecule, either its biological function or individual chemical structure. Now, the substance that is to be purified is specially and reversibly adsorbed by the ligand, and this ligand is immobilized by covalent bond on the chromatographic bed material. Okay, so chromatographic bed material have your ligand, and that is serving as the stationary phase. Now onto this stationary phase, the substance is going to be adsorbed. Okay, so the ligand is the binding substance, and this chromatographic bed material is your matrix. Now the samples are applied in favorable conditions so that those samples will specifically bind to the ligand. Now the substance of interest will consecutively bind to the ligand. Okay, the substances of interest will bind to ligand, and the unbound substances will be washed away. Okay, so you will put a mixture through the stationary phase, and in that stationary phase there are substances that you want to isolate, and there are some substances which are the impurity for you. Okay, you want to isolate from that this substance. So the substance of interest will bind to the ligand, and the unwanted unbound substances will be washed away. So that is how the affinity chromatography works. Now the recovery of this substance of interest that has been bound to the ligand is done by changing the experimental conditions, and when you change the experimental condition, then that favor the desorption of this substance from the ligand. Okay, so the recovery is done by changing the experimental conditions. So the operation of the affinity chromatography involves these steps. Okay, so the first step is the choice of the appropriate ligand. Okay, you have to choose an appropriate ligand, and then after that there is immobilization of this ligand that you chose on a support matrix. Okay, on support matrix you immobilize your ligand, and after that there is binding of the molecules of interest with the ligand. Okay, now your support matrix has the ligand, then you will pass solution through the stationary phase, and then the molecules of interest present in that mixture will bind the ligand. Okay, that's the third step, and in that step only there will be removal of non-specifically bound molecules. Okay, the molecules which will not be able to bind the ligand will be washed away. Okay, so that is how there is a removal of non-specifically bound molecules, and after this there is elution of the molecules of interest in the purified form. Okay, so the molecule of interest that has bound to the ligand, that you elute in the purified form. Okay, so this is how the affinity chromatography is done or operated. Now a biospecific ligand that can be attached to a chromatography matrix covalently, okay, that thing is one of the requirement of the successful affinity purification. Okay, you should be having a biospecific ligand that you can attach to the chromatography matrix by covalent attachment. Okay, that is the requirement of a successful affinity purification. Now the binding of this ligand to the molecule of interest should be reversible. Okay, it should be able to bind, and after that when we want to remove that molecule of interest from the ligand, then that we should be able to do. Okay, so this binding between the ligand and molecule of interest should be reversible. Okay, and if it is reversible, then that allows the removal of the molecule of interest in the active form. Now, after washing away the contaminants, the coupled ligand should retain its specific binding affinity for the molecule of interest. Okay, so after you remove your molecule of interest as well, even after that, the ligand should retain its specific binding affinity for the molecule of interest, so that that stationary phase of the affinity chromatography can be reused to isolate the other molecules as well. Now we are going to see some ligands and their target molecules or molecule of interest. So in white you have the ligand, and in green or yellow you have their molecules of interest. Okay, so if you are using enzyme as a ligand, then you will be able to isolate the substrate analog or inhibitor or cofactor. Okay, these three things can be isolated by doing affinity chromatography if you are using enzymes as the ligand. Okay, and for if the ligand is antibody, then the molecule of interest that you can isolate can be antigen. Okay, then if the ligand is lectin, then you can isolate polysaccharide, glycoprotein, cell surface receptor, and even cell as well. Okay, so these things can be isolated, purified if you are using lectin as the ligand. If you are using nucleic acid as a ligand, then you can purify the complementary base sequence and nucleic acid binding proteins. Okay, if hormone is the ligand, then you will be able to isolate receptor. If the avidin is the ligand, then you will be able to isolate biotin. Okay, if the glutathione is the ligand, then you will be able to isolate glutathione as transferase and then GST fusion proteins. Okay, if proteins A and G, okay, are the ligands, then in that experiment you will be isolating immunoglobulins. Okay, if you want to isolate polyfusion proteins, okay, his fusion proteins, or you want to isolate the native proteins for histidine, native proteins with histidine, then you will be using metal ions as the ligand. Okay, so these were the examples of the ligands, and these were the examples of the molecule of interest that you can isolate by using these ligands in the stationary phase. Now we will talk about an example. Okay, consider eukaryotic mRNA with poly A tail, and you want to separate this mRNA with poly A tail. Now for that you will use oligo-DT cellulose affinity matrix. Okay, so you have a mixture of RNAs, and in that mixture of RNA there is mRNA, and you want to separate mRNA from that mixture of RNAs. Then you will do that by using this oligo-DT cellulose affinity matrix. Now this poly A tails of the mRNA form stable interaction with the short chains of oligo-DT which are attached to the support matrix. Okay, so what is there? There is a support matrix. To that support matrix there is attachment of oligo-DT. Okay, this is your stationary phase. Now this oligo-DT has the short chains, and these short chains of oligo-DT interact with the poly A tails of the mRNA. Now, high salt in this process, while doing oligo-DT cellulose affinity chromatography, high salts are added to the chromatography buffer. And what happens because of this? Well, this results in the stabilization of nucleic acid duplexes. And why do you do this? Because there are only a few DTA base pairs which are being formed. Okay, so the base pairing between the short chains of oligo-DT and the poly A tails of the mRNA, those base pairs are very few, and that is the reason why you have to stabilize this nucleic acid duplexes, and for that you add high salt to the chromatography buffer. Then a low salt buffer is used, and when this low salt buffer is used, it is used after the non-polyadenylated RNAs have been washed out from the matrix. Okay, so like the other RNA, okay, except mRNA, when they are washed out from the matrix, because only mRNA is going to bind, okay, other RNAs are not going to bind. So when those other RNAs are removed, those other non-polyadenylated RNAs are washed out of the matrix, then you will use this low salt buffer, and the use of this low salt buffer will then release the mRNA from the oligo-DT matrix. Okay, that is how it is going to work. Okay, so that buffer helps to destabilize the double-stranded structure, and that causes the elution of the poly A mRNAs from the resin. Okay, so the double-stranded structure which was formed between the poly A tail of the mRNA and the short chains of oligo-DT, that has been destabilized by this low salt buffer, and that is how you are able to elute the poly A tail mRNAs from the resin. So this is how it looks like. Okay, this is this is the support matrix. This yellow color thing is your support matrix, and this is your short chains of DT, that is deoxythymine. Okay, and yes, this support matrix have these DT short chains, and to it you add the mixture of cytoplasmic RNAs, okay, under a hybridized condition. Now the mRNA has a poly A tail, and there is this T tail which is attached to this matrix, and that is the reason why this T tail will bind the A tail of mRNA, and all the other RNAs will be washed out. So after that you will be having mRNA attached to this oligo-DT matrix, and then you will use a low salt buffer and you will elute this mRNA from this, and that is how you do the purification of the mRNA using oligo-DT matrix. It is quite simple, but it sounds complicated. There is a matrix that has T on it, right? And your mRNA has AAAA on it, and the other RNAs don't have AAAA on them. So when you put this mRNA and the mixture of this cytoplasmic RNA into the stationary phase, okay, this stationary phase, then this AAAA will bind to the TTTT of the stationary phase, and all the other RNA will be washed out, and that is how you purify the mRNA from the mixture of the cytoplasmic RNAs. Okay, it's quite simple. Now let's talk about choice of ligand. Okay, how to choose a ligand, what are the factors, okay, for that. Okay, now this choice of suitable ligand is the most important feature of the affinity chromatography. Okay, so the factors that matter is the specificity, then reversibility, then stability, then size, then affinity. Okay, so let's see one by one. So starting with specificity. Now the ligand should recognize only the molecule of interest that is to be purified. Okay, only the molecule of interest should be recognized by the ligand. Okay, that is specificity. Now talking about reversibility, the ligand should form a reversible complex with the molecule of interest that is to be purified. Okay, the complex, okay, this ligand and the molecule complex, this complex should be reversible. So after the purification is done, if you want to remove this molecule from that ligand, then you should be able to do it, and that is the reason why reversibility is very important in affinity chromatography. Now after that, stability. Okay, now the ligand should be stable in the conditions of immobilization. Okay, you have to immobilize the ligand on the matrix. Now when you do it, that ligand should be able to bear that up. Okay, it should be stable in those conditions. So the stability of ligand and the stability of the ligand is another important feature of the affinity chromatography. Now after that, there is size. Okay, size factor. Now the ligand should be large enough such that it can contain several groups that are able to interact with the molecule of interest, and when these several groups of the ligand are able to interact with the molecule of interest, then that results in sufficient affinity. Okay, so the ligand should be large enough. Okay, it should be large enough to bear several groups on it. Okay, to have several groups on it, and when there are several groups on the ligand, then that several groups will be able to interact with the molecule of interest in a sufficient manner. And that is the reason why this thing, the large size of the ligand, results in sufficient affinity. Now let's talk about the affinity. Okay, now the binding affinity is the strength of the binding interaction between the molecule of interest and the ligand. Okay, the strength of binding interaction between these two is known as binding affinity. Now this binding affinity is measured in terms of equilibrium dissociation constant, and this equilibrium dissociation constant is represented by Kd. Now smaller the Kd value, the greater will be the binding affinity between the ligand and the target. Okay, ligand will bind with more force to the target if the Kd value is small. Okay, the smaller Kd value, greater is the binding affinity between ligand and target. Now this equilibrium dissociation constant is inverse of equilibrium association constant. Okay, now this equilibrium association constant is represented by Ka, and equilibrium dissociation constant is represented by Kd. So Kd is just inverse of Ka, or Ka is just inverse of Kd. Now this reaction is representing the interaction between the molecule of interest M and the ligand. Okay, M will interact with L, and this Ml complex will be formed. Now the equilibrium association constant and the equilibrium dissociation constant of this reaction is given by this equation. So Kd is equal to the concentration of the molecule into concentration of ligand divided by the concentration of their complex, and Ka is equal to concentration of the complex of the molecule and ligand divided by the concentration of the molecule into the concentration of ligand. Okay, so this equation is giving you the value of equilibrium association constant, and this equation is giving you equilibrium dissociation constant, and these two, the value are in accordance with this equation. Okay, so that's all for this video. Thanks for watching.